It's always, makes me blush, because are they going to work or are they not?
Yes.
Can I get my computer connected even?
Right.
Oh, I don't know if my audio's on and they can hear me, or
Yes.
if that's for here. Yeah, because it
Yeah, this is for the
Zoom.
yeah,
Okay.
streaming.
Yep. Okay. Hola.
Francisco, what to decide?
Francisco, introduce.
Bueno, pues muy buenas tardes a todos y a todas. Pues vamos a empezar con esta plática y primero quiero presentarles a nuestro invitado, el doctor Walter Besio, que es profesor del Departamento de Ingeniería Eléctrica y de Computación y es Departamento de Ingeniería Eléctrica, Computación y Biomédica de la Universidad de Rhode Island en Kingston. Y pues además es empresario, tiene la empresa CREmedical, que es el fundador y el principal investigador de la empresa. Una reseña muchas gracias. Perfecto. Una reseña muy breve de Walter Besio. Es la siguiente. Él estudió la licenciatura en la Universidad de Florida Central y luego la maestría y el doctorado en la Universidad de Miami. Estuvo trabajando durante 12 años en la industria antes de incorporarse a la academia. En esa industria estuvo, digamos, involucrado en electrónica y equipo biomédico. O sea, su experiencia en la industria es en electrónica y dispositivos médicos. Se especializa en el desarrollo innovador de instrumentación biomédica para el diagnóstico y las terapias que mejoren la calidad de vida de las personas. Sobre todo en todo lo relacionado a la enfermedad y a la discapacidad neurológica. Su trabajo involucra de manera muy particular una invención que tiene Walter de hace algunos años, que es el de los electrodos tripolares de anillos concéntricos. Entonces, todo lo que él ha desarrollado en los últimos años ha sido enfocado a las aplicaciones de estos electrodos tripolares de anillos concéntricos y sobre todo en cardiología y en neurología. Particularmente dentro de la neurología, su tema más importante o la investigación más importante es alrededor de la epilepsia. Y de hecho, por eso el doctor Besio fundó también o fue cofundador en la Universidad de Rhode Island del programa interdisciplinar de neurociencias. Salud. Además, bueno, pues el doctor Besio es miembro del Instituto de Ingenieros Eléctricos y Electrónicos y tiene muchas otras membresías dentro del instituto también en la Sociedad de Medicina y Biología y muchas otras. Ha sido colaborador del IEEE, como se llama el instituto, en varias áreas del IEEE y fundamentalmente en la Sociedad de Ingeniería en Medicina y Biología. Bueno, pues yo creo que sigue acá su resumen curricular, tiene más de 50 publicaciones, varias patentes, yo creo que más o menos como una docena de patentes. Es un inventor y un desarrollador y un investigador bastante prolífico. Entonces, bueno, démosle la bienvenida al doctor Besio con un aplauso, por favor.
Thank you for coming today. I'm honored to be here. I'm really happy to be here. Viva Mexico, viva los mexicanos. Something like that. Yeah.
If you can
Move over here.
Yeah, so that they can see.
Yeah, very good. Okay. So hopefully my voice will be loud enough that you can hear me. Oh, I brought some show and tell. I don't know if you guys have show and tell, when you're in kindergarten, but I always enjoyed bringing things like frogs and grasshoppers. So I have some things to show and tell today. This is a regular disc electrode. This is what everybody is using trying to get signals from the brain. And then these are tripolar electrodes. And so I'll let you see what they're like. And we'll pass these here and these ones over here. Okay, so I've got 3 hours, I heard. Just kidding. But I have a lot of slides and hopefully, that you'll be able to understand. But like I said earlier, at any point that you don't understand me, just, raise your hand, say, please stop or anything so we can get on with it. Okay, so a little background in electroencephalography, which is getting the signals, electrical signals of the brain. It's nearly 100 years ago when Hans Berger did the first recordings in humans of EEG, not from outside, but from inside the brain to get inside the skull to get the signals. But he first saw the waves that we're still getting now. And the electrodes that he used were basic disc electrodes, like the disc electrode that's going around, not tripolars, but like disc electrodes. And so it's nearly 100 years, everybody's still using the same electrodes. And where do the signals come from? So, let me see. This, does it work here? No. Well, anyways, pyramidal cells in the cortex, they're pretty much lined up in parallel. And so when they get activated, if you activate many of them, they sum together. Oh, what is that principle that you guys have that's summation, basically, that you can look at things one at a time, but they all get summed together? Superposition. Do you remember that? So that's how this works, basically, that there's many neurons that get to fire together in unison or near unison, so we can detect those signals on the scalp surface. And these are some of the waves. The slowest waves are delta waves, and then theta waves, alpha waves. Alpha waves you typically get when you close your eyes. And then in the occipital areas, they become very strong. It's a sign of relaxation, is what's considered. And then beta waves are 14 to 30 hertz. Not too many years ago, back at the beginning of when I started working on this in like 2002, people thought that the fastest waves of the brain were 30 hertz. And one of my mentors, Bob Fisher, who's the head of the epilepsy department in Stanford, was the first to publish that there were faster frequencies prior to seizures from the human brain. And so beyond this is the gamma band, which is 30 hertz to about 80 hertz, then high gamma, then ripples. And we're going to show you some signals that we've gotten that are all the way up near 500 hertz prior to people having seizures. Okay, so now this part this kind of goes off into a little different direction here. That how did I get to have all the fun things that I get to do? As a kid, I used to dream about traveling, but grew up, from very in a poor area, I guess you might say, with poor parents, and never thought that I'd get a chance to travel. But somehow, as a researcher, this has changed. But my father, he was a machinist, used milling machines, lathes, things of that sort to build things. And he would get plans from engineers, and my dad would come home complaining. Those engineers have no idea what they're doing. They went to college for nothing. It's a waste. They give me these diagrams and there's no way that they can be made or that they'll ever work. And they have no experience and things of that sort. And so from the beginning, there was no way I was going to be able to become an engineer, according to him, that experiential learning was the way. Okay, so the reason I put my mom up here, when I was still in her womb, she was told that she had cancer and that she could get treatments for the cancer, but if she did that, her baby would not survive. And so this woman made the decision that I would be the one that would go on. And so that's pretty much where I get my inspiration to keep working to do the things that I do, that I have to keep pushing to do more. And so then this education, how many of you came straight from high school into college? Most of you, right? Yeah. So I kind of started that way. However, I wasn't planning to go to college. I was actually planning to become an electrician, somebody who connects wires in your house, right? You know, wiring houses. I was going to school in high school for vocational school. And because, you know, like I said, my dad was like, you're not going to college. And so, but eventually, a guidance counselor in high school said, oh, you need to go take this test. How am I going to get there to take that test? Oh, so then he grabbed one of my friends and said, you're going to go take this test on Saturday morning and you're taking Besio with you. And so I got to go take a test that's a college entrance exam test. And that got me into a junior college certificate program. And so then I was going to work in that, which was like industrial wiring. And then from there, I was going to go on for 2 more years to do engineering technology and get an associate's degree. Then go on to university to get 2 more years in electrical engineering. Well, after my first year, I decided that I was living in northern New York, and we wanted to ride our motorcycles to Florida for spring break. Well, in northern New York in the spring, when our spring break was, it's still ice and snow all over the ground, and there's no riding motorcycles. But eventually, I did leave school, which I don't recommend this for any of you, and go and work on a dairy farm, okay? And I worked on a dairy farm for a couple of years and, you know, learned about taking care of cows and all the equipment and things of that sort. And then realized, I should go back to school. And so I went and checked out a college job fair type thing and checked many different universities and schools. And one of these little technical schools guaranteed me that I would get a job after graduating from them. Not only would I get a job, I would get many offers of jobs. And so then I went and talked to other universities about it. None of them could guarantee me a job. And so I was like, okay, you know, I'm practical. I'm going to go to this technical school. And so I went to this 2-year technical school, had, I think, 27 job offers when I got done with it to become a technician, electronics technician. and started working in a research and development department in an electronics company, and then wanted to go back to school and found out that my 2-year degree that I got from that school was not accredited. Fortunately, the degree you're getting here is accredited. And so I had to start all over again to get my associate's degree, and started again going to junior college after work, and got my associate's degree. Then went on to the University of Central Florida to get my bachelor's in electrical engineering. And then I had a friend who was, he was an engineering student who came and did an internship with me when I was working. And when he graduated, I went back to school full time. We switched places. He worked in the company as an engineer. I went back to school full time. And then I just wanted to get my bachelor's and get done. But he convinced me, he went on to get his master's, and he convinced me, oh, Walt, it's just a couple more years. You ought to go get your master's. And so then I went on to get my master's at the University of Miami, and worked at the Miami Project to Cure Paralysis with people that were paralyzed to work on my master's. But my master's wasn't a 2-year degree. That thing was 4 years to try to get me done, because my advisor got kicked out of the university. And then I was left with a committee and trying to get done with a committee, going around to every one of them, and they all wanted me to do something different. And then I stayed and did he went on to get his PhD and convinced me, oh, if you really want a good job or you want to do something interesting, you need to get your PhD. So then I went on to get my PhD. And then I was like, okay, all I want to do is go back to industry. And then I realized, oh, if I want to keep working on the technology that I'm working on, I can't go to industry. I've got to stay in academia. And in academia, they call it publish or perish, which really means you got to beg all the time for funding, money, and so you can pay your students, and you can pay technicians, and you can buy supplies and things like that. So that's what that's all about. So that is how I got where I am. And this is another person who got me into it. This was my older brother, who was in an automobile accident when he was 25 years old. He was as I was talking about, northern New York, we have a lot of snow. And so his truck had stalled, and he was on the side of the road, but couldn't get off the road because of big snowbanks. And so he was looking under the hood, trying to figure out what was wrong with the engine, why it wouldn't start. And somebody came along from behind, hit the truck, and the truck went over the top of him and crushed his leg and spinal cord. And so he was paralyzed from the neck down, C3, C4. So that's what got me into all of this. And so, okay. So now, I never used to think that one person could make a difference anywhere, basically, other than just in your own life. But when I first moved to the University of Rhode Island, I use electrical stimulation to try to alter the brain state. And so I needed to find somebody, as an engineer, I'm not going to be cutting brains and slicing them and coloring them and all those kind of things. That's not what I studied. I studied instrumentation, bioinstrumentation. And so I needed to find somebody that could help me prove that I wasn't burning brains. And so I went all over the University of Rhode Island trying to find somebody who was doing things or had those techniques. And I found many researchers that were doing neuroscience research, mostly on fishes and crustaceans, lobsters and things of that sort. and then finally found one person who was working in Alzheimer's who was actually doing similar to what I wanted to do. And so, but when I met him and told him about all these people that are doing neuroscience, he had the idea, maybe we should start a neuroscience program. And so we started the graduate program in neuroscience there on our own. And then I don't know if you guys, I don't think you have CVS pharmacies down here, but CVS is one of the largest pharmacies in the United States. And one of our graduates from University of Rhode Island is Tom Ryan, who was the president CEO of CVS. And his dad died from Alzheimer's. And so he was trying to find some place to invest in to set up a center to study neurological disorders, primarily Alzheimer's, but wanted some place that could make a difference. And so he went all over in Europe and all over the United States and other places to try to see where he could invest his money. And when he saw our program, that we had engineering in the neuroscience, even though it's not really science, but it is applied science, he thought, okay, this is practical. Instead of just doing papers and publishing and getting people out, they have engineering and they might actually make products that can help go out to help people. And so he gave us a bunch of money to start the Ryan Institute. And then recently, 3 years ago, we started an undergraduate program in interdisciplinary neuroscience as well. And then also, when I first moved to University of Rhode Island, we had these crummy, terrible buildings, and the labs were just terrible. And in 2008, when I moved there, there was a new president, President Obama, and we had a recession at that time, and he had stimulus money. And part of that was you could get money for biomedical engineering buildings. And so I wrote a proposal to get money to improve our biomedical engineering department buildings. And the researcher, well, my department liked it, my college liked it, but the research office said, there's no way you can submit. Biomedical engineering, they laughed at me and said, the College of Engineering isn't even on a master plan. I had no idea what a master plan was at that point, but apparently it's something about how the university makes buildings. And but pharmacy was going to be submitting the proposal. And so I was able to help pharmacy get their building. But my chair, or not my chair, but the dean of the College of Engineering kept pushing and eventually got the university to approve us. And then the state legislators, and then it went on to a ballot to get $450 million for a bond so we could build this building. And they voted for it, and now that's the new building we have. So that's kind of why I'm telling you, the sky is the limit. Whatever you can imagine, you can do. Okay? So now it's for your math. Okay, so as you've seen, regular disc electrode and tripolar electrode, same size, but in this one we take the differences. And this is just, if you think of these red dots as electrodes on somebody, and they're getting potentials. And so if you take the difference between, we'll say, V1 and V0, and V3 and V0, you get two differences in the x-axis. And then if you take the difference between V2 and V0, V4 and V0, you get two differences in the y-axis. And so Do you guys remember what Laplace is? Second spatial derivative. So that's the two differences in the x and y in this case on the surface. And so that is done with the 5 points, and then it's done again with the 9 points. The 5 points had already been done. And to get your PhD, you can't do what other people have already done. So I had to complicate things and added those extra points and worked out the equations for that. And if you do the Taylor series expansion on these points, this is what you end up with after you do a bunch of cancellations. And so that is our Laplacian algorithm that we put into our electronics, which is here. So this is a preamplifier that I've designed to get small signals. So from those disc electrodes, the typical EEG signal is 1 to 10 microvolts. Okay. Now, what did you see with those tripolar electrodes? How close do you think they are together? They're about 1 millimeter apart. Okay. And so with regular disc electrodes, they're typically, the minimum you can put them is 3 centimeters apart. Otherwise, they'll see the same signal. Okay, but 3 centimeters apart. These ones are 1 millimeter, a little less than 1 millimeter. So what do you think that does to the intensity of the signal that we record with the ones that are real close together? Does it make it bigger or does it make it smaller? So because they're getting signals from a much smaller area, then they get a much smaller signal. Instead of microvolts, we're getting nanovolt signals. And for those of you doing like common mode rejection and amplifier gain and things of that sort, that's in the noise of most EEG amplifiers. Like this amplifier here, the noise level for that is 1 microvolt. Well, no, excuse me. I take that back. It's not 1 microvolt. It's, 100 nanovolts, 0.1 microvolt. Okay. And our signals are like 200 nanovolts. So that's just barely above the noise level in those, and using those electrodes with those amplifiers, even the best amplifiers that are out, you don't see any signal. And so that's why we had to develop the electronics for it. Okay, let me see if I can get this to work. Okay, so on the back of the head, we've got 6 tripolar electrodes. Okay, feel the back of your head. Do you feel all those muscles there? So when you're trying to get signals from the occipital lobes, all those muscles are generating gigantic EMG, which is millivolts, hundreds of millivolts. And so the top 6 are tripolar signals. The bottom 6 are regular EEG signals. And she's going to turn her head, which is going to activate those muscles, and she's going to close her eyes. Regular EEG, all that black, that's the muscle activity. That's electromyogram. But if you see these waves here, that's her alpha waves from closing her eyes. There's no signal processing done there. That's all just because of the way the electrode is designed, that those little electrodes are only 1 millimeter apart. Okay, let me see if I can get it to keep going. Oh, okay. And so these are at the beginning of seizures. Regular electrodes, tripolar electrodes. These electrodes are right next to these electrodes. This is muscle activity as well here. but yet you can see clear brain activity here. Onset of another seizure, and I like this touch board. So this is all muscle activity at the onset of the seizure. And the epileptologists, neurologists that are trying to see what's going on, where those seizures are originating, in that muscle activity, they can't tell. And so our system, you can see EMG there, but it's much, much smaller because for us, the EMG is a global signal coming from far away. And we only focus on what's directly below it. And the EMG is making nearly the same potential on each of the electrodes. And when you take that, well, I'll show you. Okay. So for those of, oh, have any of you been to like Wet and Wild or water parks? You know, so you have the water and then your tube that you can float down the river on, the lazy river or whatever. So if you think about the pond of water and then the breeze blowing and it generates ripples on the water, okay? But inside this tube, those little ripples and waves are flat. It's nice and flat, like glass in there, we say. And so inside here is like where our middle ring and center disc are. This acts like our outer ring, which acts like a shield to all that noise and chop coming from far away. And then inside, it's really clean. And if you remember that formula from previously, the middle ring minus disc gets weighted 16 times more than the outer ring and center disc difference because it has more information about the local sources there. And then since you're engineers, and not only that, you're bioengineers, you know, so here's the circuitry, difference amplifiers. And on the electrode, if you think about this lime green is like your AC main signal generating nearly the same potential on each electrode. And then if you have high common mode rejection, then that AC mains gets canceled. And the difference signal comes through. So it's really very simple. And that's the way I try to keep everything. You know, that thing quote that's keep it simple, stupid. Sorry. But that's the way you do it to make it so that it cancels out all the global noise and you get a much better signal to noise ratio. Okay, another nice video. Okay, so he has the electrodes on the back of his head again. Visual area, visual cortex, occipital lobes. Checkerboard pattern is going to reverse what's black to what's white. And it keeps doing that. It's going to change 10 times. And each time it does that, it generates visual evoked potentials in the occipital cortex. And so that shows where they are. And once again, it's going to show up that the top 6 are tripolar, bottom 6 are the regular EEG. Every time a stimulus is sent, that it changes the checkerboard, it sends a trigger to the amplifier, and we're going to use that to window 1 second here, or actually 500 milliseconds of the signal centered around that trigger. and then we're going to average them, ensemble average them. And so in real time, you'll see tripolar ensemble average, regular EEG ensemble average when this starts flipping. 1, 2, 3, 4. Within 4, we had a good signal to noise ratio showing the visually evoked potential in the tripolar. In the regular EEG, there's something there, but if I didn't tell you that was there, it probably just looked like noise. Okay, so that's how much better we get for signal to noise ratio. And it'll take at least 100 with regular EEG stimuli. So one of the other areas that I did the work in is brain-computer interfacing. You know, everything I do is brain-computer interfacing, but in this case, this one is for imagined movements. You think about moving parts of your body. And in this case, it was think about moving your left hand or think about moving your right hand. And this is showing how a very simple way of determining, is it correlated with moving the left or correlated with moving the right? And so the darker this is, the higher it's correlated. And it tells us which electrode. These are regular electrodes. These are the tripolar electrodes. And so between 10 and 12 hertz on that electrode, which is C1. So C1 is the primary motor cortex for your right hand, right side. And so when we see that power in that electrode at that frequency, we know they're thinking about moving their right hand. And then for left, it would be the other way, that we'd see something in C2. And so then these were people that have never done imagined movements before. And so they tried to do it with regular EEG, which is the blue, and that's the percent correct, getting up to about 60-some%. And then with the tripolar, over this one session, they got to around 85%, something like that, with the imagined movements. Now, though, we've actually started using convolutional neural networks, your AI, to study the signals. We don't even do any processing on them. And now we're doing 10 digits and letting the convolutional neural network look at the raw signals coming in. And with just a single electrode, we can get all 10 digits of imagined movements about 99% of the time. So if you have to have a hearing test, in particular for babies, they end up with getting auditory brainstem response to see if it's working properly. Typically, this is done in an anechoic chamber where it's really quiet. We do these kind of things. Everything we do is just in our noisy lab. And so within 100 chirps, which are the audio stimulus, we can see all the waves that are needed for the audiogram. But regular EEG, there's almost nothing there. It takes 1,000 to 2,000 stimuli to get the brainstem response. Okay. So then this is 2 electrodes on this student that volunteered. And she's trying to hit the targets, just thinking left and right, up and down to hit the target, which that brought the robot arm down. And now it's going to turn the robot arm clockwise. It's not a great video, but you know, it's the best I've got for it. but I can't take that giant robot arm to bring it here to show you, right? So I've got students working on a little robot arm now to control it. So then this now, when she hits this, it's going to turn it back counterclockwise. So that's just her thoughts, that she's moving the cursor to control this. Okay, so then a lot of my work is in epilepsy, working with the epileptologists. This top trace is a bipolar signal from where the seizures are originating. Why do they do bipolar? Because it helps get rid of noise. But like I said, they have to be multiple centimeters apart. And so they still pick up lots of noise. This is a time frequency analysis spectrogram. And so frequency is increasing, time is increasing that way. Partial seizure is an electronic seizure. You can't tell that the person is having a seizure. In their brain, there's electrical activity that's showing there's a seizure, but from outside, you can't tell, other than they might be just staring off in the distance, something of that sort. And then the generalized seizure is what they call a clinical seizure, when the body starts moving because it's going through the motor cortex. And so this is a tripolar electrode close to one of these electrodes. And what we found was 2 minutes before the partial seizure, we started seeing these little pulses showing up in this electrode that told us that the person was about to have a seizure. We've seen this in 14 patients that we've recorded and gotten seizures with regular electrodes and tripolar electrodes at the same time. And this is another one, similarly, regular bipolar signals and the spectrogram for it and our tripolar sensor. And then I'm going to expand this tiny little part here, look at the spectrogram for the regular EEG, nothing in it. Look at the spectrogram for the tripolar. It's pretty obvious, right? You see those dots or those pulses? The person's brain is having these narrowband, high-power pulses quite regular. And then if you expand it out, you can actually see it on the time series trace. That's what we call high-frequency oscillations. And so that's what we've been seeing in every patient that we've been able to record. And then one of my students took the MRIs from those patients and made realistic head models, and then took the high frequency oscillations and localized them to where they would be coming from. And this is what 4 of those that he got at these different frequencies. And this one, actually, we saw this starting 4 hours. Oh, actually, it's this one that, yeah, this one started 4 hours before at 425 hertz. So, and this is 200 milliseconds of those high frequency oscillations where he localized them. And so it's in the visual cortex here that it's showing up. So the neurosurgeon can see where it is, so they can decide if that's a place that they can actually perform surgery. How many of you have looked up into the sky when there's a thunderstorm and see the lightning bouncing around in the clouds and things, right? So watch this, and tell me if it resembles a storm in this person's brain. Does that look like a thunderstorm going on in their brain? Yeah. And not only that, when they had their clinical seizure, the generalized seizure, it showed up on the other side, on the right side. So the clinicians thought that it was originating from the right side. But in this patient, we saw it start on the left side 4 hours before they had their clinical seizure. And so we told them that they might want to look on the left side for something. And when they went back and looked at the MRIs, they saw that they actually had a small lesion, a small white spot that's considered like a cut in the mesial temporal lobe. Okay, so how many of you like octopuses? They're really smart. They're mollusks, and they're really smart. And so this is part of the fun that, as a researcher developing instrumentation, I get to work on all kinds of cool things and working with octopuses. I had a very large grant back a few years, and there were only a few in the US. And another person got one as well. And at the Society for Neuroscience program, he had sent out to the few principal investigators of those awards to see if anybody wanted to collaborate. And so I was the only one that showed up. And we decided to work on a proposal together. And it was going to be about driving, looking at the stress from EEG while somebody was in a driving simulator. And because they get a lot of muscle artifact from moving and things. So we decided to work on that. 2 days before the proposal was due, he emailed me saying, can you record signals in salt water? And one of my first publications, we showed that we were recording signals in salt water. So I said, yes. Well, we threw out the proposal and switched it to, let's record from octopuses in salt water and see if we can get their brain activity. And lo and behold, the proposal was funded because it was so bizarre. It's like these creatures are the most remote, alien to human brains. However, their computing power is amazing. They have as many neurons as like bears, lions, cats, dogs. They can learn from watching you. As a matter of fact, they know when I come to the lab, and they only see me once in a while. And some of them are playful, like they'll spit at you and things like that. It's like, oh, you're the guy that's going to put me in that little chamber. And so it's like, you get anywhere in there and it's, yeah. And so they'll try to get away from me as well. But what we did, one of my PhD students put together these arrays of tripolar electrodes. We put one underneath them, put one on top of them, and then with fiber optic probes, we sent light from an LED that is in the range that they're supposed to be able to see, and then provided that at different frequencies, and we were recording to see, could we get a visual evoked potential from them? You remember we had the checkerboard and we saw that visual evoked potential from the guy. Well, we tried it that way initially, but we got way too many artifacts using a display like that. So that's when we had to switch to the fiber optic probes. And this shows us that this is one frequency that we're using here, and we can see this is the visual evoked potential at that frequency on this electrode. And then there's some muscle artifact that we see on this electrode. And then, so if there's muscle artifact that we see from the tentacles, what do you think that means? That means they're moving, okay? And so what we saw was they moved from wherever they were that we were getting the signals here. And then we started picking up the signal on a different electrode. And then it stayed there. And then we picked up more artifact. And then it went to another one. And so we just submitted this, this work to PNAS. I don't remember what that stands for, but it's nature. Okay, so that was recordings. And so then a lot of my work has been stimulating, which is instead of detecting signals, we're injecting signals. And oops, this is my best project. I thought I deleted that slide, but I guess not. But anyways, that's my daughter from many, many years ago. So computer modeling of the stimulation in a 4 concentric sphere with electrodes the same. These are disc electrodes with the same area as tripolar electrodes. And white means that the brain would be activated. So in this case, it's kind of like frying the brain. And then here, we're just stimulating just a little, activating a small part of it. And we also did computer modeling to see what temperature we would get. So if the skin gets over 44 degrees C, it could possibly start deteriorating. So our computer model told us the maximum we were going to get to was 38.6 degrees C. And when we did the actual experiments, we saw that it was below 39 degrees. And I couldn't believe it. So I made the students do this over and over and over. And then I come to believe, wow, those models are really quite valid, even though they're quite simple. And then we did the tissue analysis. Remember, I went all over trying to find people that could help me do the tissue analysis. And so the scalp was fine when we did it at a certain intensity. The cortex and the hippocampus were okay. And, oh, but reviewers, remember I was saying that I'm always going around with an open hand begging for money? Well, so the way I beg is to the National Institute of Health and to the National Science Foundation. And to do that, you have to write proposals. And so all of those things I was showing that it was safe, well, they kept coming back saying, oh, you showed it was on the scalp. Oh, you showed it on the cortex. Oh, you need to show something more. And so then after I showed all of this, they said, oh, that's all static. You need to show that you're protecting the brain in its active brain. So that we did this novel object or recognition test where there's a box that we put 2 objects in, and then the animal goes in, you give them time, and they get they get familiarized with the 2. And then you take them out, stimulate them, put them back in. And the hypothesis is that they're going to spend more time with the novel one. And so if they spent more time, equal time, then we messed up their brain. But we didn't. And in humans, we've shown that we're not messing those brains up either. And we've used, do you know what penicillin is? So don't use penicillin G. It could give you seizures. But nowadays, we don't use penicillin G. Pilocarpine is really a strong type of seizure that causes status epilepticus. In the United States, 20 to 40,000 people die each year from status epilepticus. So we were proving that we could stop status epilepticus. Drugs don't work on it after 5 minutes, and it really eats the brain. And so we showed that even 15 minutes in, we could stop them. And then another model called pentyletrotetrazole. I just say PTZ. Turn the stimulation on, it stops the seizure. Not turning the stimulation on, it keeps going. And how many of you have rats home as pets that have seizures? Any of you have rats for pets? That's close. So I was going to say we developed an automated seizure control system for rats, you know, in case anybody has a pet rat that has seizures. But we really didn't develop it for rats. We developed it for humans, but we had to test it in rats. Okay. So, and what this is showing is that we can detect a seizure, and in these are controls, we don't do anything. In this group, we detected the seizure, gave one dose automatically of the stimulation on the scalp, and it stopped the seizures. And then in this other group, one treatment didn't work, and it detected another seizure and applied a second dose, and that stopped the seizures. And then I always liked, when I was reviewing papers, I always liked these graphs. I thought they were very beautiful. And so, you know, I got a colleague here in Mexico at ISS that she does this. So she looked at our brains and made one of these beautiful figures for me that shows that we protect the brain, and diazepam can be potentiated. You can use a sub-effective dose, and if you apply the stimulation, it makes it much more effective. Why is that important? Well, the anti-seizure drugs what would you say? They slow you down. And so what we want to do is minimize those side effects. And so lowering the dose and making it more effective is what we're trying to do. And in status epilepticus, the black ones show that glutamate is constantly increasing, which is feed forward. For those of you thinking of, you know, today you were looking at feedback for those amplifiers. This is feed forward, you know, that the more glutamate there is, the more excitation there is, okay? And, oh, but when we stimulate, the glutamate stays down, so we're stopping it. Okay, and these ones, that's another model that we used that worked. And P-glycoprotein. I have no idea what it is, but my colleagues do. And what happens when somebody has a seizure, P-glycoprotein comes around and gets into the blood-brain barrier, and then the anti-seizure drugs can't get into the brain, and they don't work as effectively. And so what we found was that if we stimulate after there's seizures, we prevent the P-glycoprotein from forming so that the anti-seizure drugs will still work. And then this is drug companies test on this model to see if their drugs can help stop the brain from becoming epileptic. And what the takeaway here is that after 26 days, all of the controls had become epileptic. But in the ones that we stimulated right after the stimulation in the brain from outside, they were still in stage 2. Nice. Which was just like eye blinking. And at 40 days, they still hadn't gotten out of that. However, the master's student who was working on this wanted to graduate that semester. And so to get done, we said, okay, stop giving the TFS now and keep giving the internal stimulation. And so you can see that after we turned off the external stimulation, the brains were protected in these animals for 20 more days, that they didn't move out of that. And it took them 50 days to become epileptic in total, which is twice the time that it took for the others. And more of the P-glycoprotein. More head models to show what the stimulation is doing. This is more like TDCS, where they have 2 large electrodes far apart. It's acting on both areas. When we stimulate, we increase GABA, which is the major inhibitory neurotransmitter, and decrease glutamate, which is the major excitatory. In epilepsy, there's too much glutamate, not enough GABA. So how do you say lucky? Sueño? Yeah. So we got really lucky here. Mucho sueño. Suerte. Okay. Yeah. Yes. We're sleepy now. Okay. So then we're looking at genes as well. And, what we're seeing is that in dopamine, we're protecting certain pathways for dopamine, which is what the problem is in Parkinson's. And so there's a very good chance that we can help in that as well. And this was a model of Parkinson's that we showed that we can protect those brains, but the genes help us understand the pathway, blocks pain, very focal when you stimulate. And so those are the takeaways. And lots of collaborators, lots of places that when I was begging for funding, we got funding, and lots of places using our devices. And that's what the things look like for recording, and that's the stimulator. Preguntas.
Bueno, si alguien tiene preguntas, pasarle el micrófono.
Sorry, I went over. But I thought I had 3 hours. Yeah.
A ver.
For Parkinson's disease. ¿La repito? Sí. Can this approach can be used for Parkinson's disease? For helping it?
We haven't proven that yet. We're still trying to prove that we can use this for epilepsy patients. And that's actually part of why I'm here now, is we're treating patients with epilepsy. But the animal models that we've shown so far and the genetic work says that it should be consequential in Parkinson's disease. Not only Parkinson's disease, but okay, so you saw that we decrease glutamate as one of the things that I just saw. So in the past, people working on Alzheimer's, their thought was there were plaques and tangles. Let me think, what are those other things called? Amygdala, no, not amygdalas, but well, I'm an engineer. Okay. So they show all these bio things that are in the brains of people with Alzheimer's. But over like about the last 5 years, researchers are starting to show that it's glutamate toxicity in Alzheimer's, Parkinson's, and epilepsy. So since we're lowering glutamate, there's a very good chance. Plus, what we showed was we're able to help protect the dopaminergic system. So there is a chance. The problem I've got is I'm just one person, and I need more funding and a whole team. Good question. Any other questions?
When applying the electrolyte, don't the outer rings and the inner rings get shortened?
Excellent question. And that's when everybody talks about putting electrodes on the scalp. If they get too close together, they say it's salt bridges, shorting out the signal. That's what happens with regular disc electrodes. What we have in that 1 millimeter space is we have about 2 k-ohms of impedance. And that is so small, but when the ionic currents flow up into the scalp and through the paste, they generate currents that then are converted from ionic currents to electrical currents that we can detect with our circuitry. They're only generating, like a couple hundred nanovolts though, over that 2K ohms. But it's enough that we can see them, amplify them, so that amplifiers can see them afterwards. Yeah, excellent. Any other questions?
Okay. First of all, thank you very much for the interesting presentation and for showing us all the work you've been doing over the course of many years. I think it was really interesting and eye-opening for a lot of students here. So we have a lot of biomedical students just here with us. So how can they start collaborating with you? Are there some opportunities? Also for our researchers here, how can we start maybe some collaborations going?
Definitely. Yeah. So when you want to do your projects, to graduate, you're all welcome to come to Rhode Island. That's one thing. But alternatively, if you want to stay here, we can do projects. So Jose Luis has one of our the system that you saw those videos from, that's actually, there's one of those here. And we can get more systems here, if there's people interested in doing collaborations and in working on continuing these projects. Excellent. Yeah. If you can't ask the question in English, don't worry. Say it in Spanish. Somebody will give it to me in English. I know what it's like. It's really hard for me to try to talk to somebody in Spanish, even the simplest things, so don't worry.
First of all, sir, thank you. And well, my question is, what other applications do we have for these electrodes? Well, we have seen that they have improved the signal, but what else can we do with this? Or are they exclusive for EEG? Thank you.
Yeah. So there's many other areas that the therapeutic part, I think, can go, such as Parkinson's, Alzheimer's, Huntington's, psychological disorders like depression, schizophrenia. Oh, so there's a group in Australia who has one of our systems, and they study schizophrenia. And what they were doing, they had animal models that they showed that they could see high frequency signals from schizophrenic brains, but they couldn't see those in humans that they knew had schizophrenia. But they were working with doctors, and so the doctors gave muscle relaxants to the patients. And then, because the EMG was gone, they were able to see the signals, the high frequency signals from schizophrenia. And so they went on doing a lot of signal processing and using 128 electrodes, and came up with an algorithm to minimize the EMG so they could see the high frequency oscillations. And then they found out about our system and started using it. And they've just submitted a publication for review where one of our electrodes gets them everything they need. Instead of all the signal processing, it just automatically shows them whether the person has schizophrenia or not, and whether the drugs are helping to lower the schizophrenia. Not only does it help, it actually shows that in healthy and in schizophrenia, the drugs that they're using only get rid of part of what's going on in the brain. There's still, when I look at the signals that they're getting, there's still something else left that the drugs are not minimizing. So that's one of the areas. And then because somebody else there saw how good the signals were working and looking. They're in sleep studies. So they got a system for themselves. And they've found that in sleep studies, they're much, much better because they don't get all the EMG and all the movement artifacts. And they've just submitted a publication now for review showing that as well. And then, of course, there's fetal ECG and fetal EEG, and even EMG. We can see the local motor activity in our EMG. Rather than all that, just black garbage that you pick up, we can see the motor units firing and things. So there's many other areas to use for recording, and stimulation, the therapeutics. Excellent question. Yeah.
So I think there's no more questions. So I just would like to give you, on behalf of the university, a small recognition for this really great presentation that you just gave us. Thank you very much, and we hope to have you back here at the university. Thank you.
I have this one. Thank you.
Thank you.
Yeah. Gracias a ti.
Bueno, muchas gracias a todos por acompañarnos y pues nos vemos en la siguiente presentación y seminario. Muchas gracias.
Gracias.
Y si tienen más preguntas, aquí va a estar Walter todavía, un par de minutos, ¿no?
No, really. Gracias a ti. It's like, I enjoy it.